This is the second week of our Microbial Omics seminar series for beginners. And just some housekeeping business first that I'd like to take care of. As a reminder, the purpose of this seminar series is to introduce some of the key concepts and computational strategies behind popular omics approaches to at a level that is as simple as possible, but without trivializing these concepts as much as possible. Each session is supposed to have a seminar part, which is now what's happening here, that should take about an hour, and the discussion part that should take about 45 minutes. Again, seminars will not cover tools or best practices, but the discussion part will be led by your questions, and we will discuss things as a community. So then we can talk about tools and best practices, whatever comes to our way. Last week, we had some learnings. We use Zoom for those of you who are here, and I also would like to welcome those of you who are joining us through our live YouTube stream. It was very difficult to follow questions on the chat window, and many questions were left unanswered despite our best intentions. And this is partially due to perhaps not very effectively using chat window, because there was a lot of in-between discussions that were not related to questions. But also the fact that we have not been fast enough to catch your questions, and we basically lost connection with you, and we're not able to answer them later. And YouTube live stream was, things did work much better than I expected, actually. Things went smoothly, and I hope this week it's going to be somewhat similar to that as well. But also on YouTube, we lost all the live comments and questions. So those questions that came as during the live session, although we did our best to respond immediately, once the session was over, they were lost. So we couldn't get back to those questions that we could not answer. But we're not going to let it go. This week, again, we have Eva on Zoom call, who is going to try to respond to your questions on the chat window, along with other members of the community, just like last week, because I'm very thankful for those of you who tried to help participants by answering their questions. And we have Emily on the YouTube side, who is going to try to respond to questions on live chat on YouTube. And this week, we improved the situation a little bit, hopefully. We added Andrea Watson, who is also a graduate student at the University of Chicago. She is going to carry questions from the YouTube environment and Zoom chat environment by surveying your questions, and add those questions that are important for others to hear about or have an answer for later. And she will add them as comments to the YouTube video. So we identified that the YouTube video should be the primary source for this content and the questions that follow. So people later, for instance, watching this content could perhaps find more resources to understand concepts that may not be clear to them. So basically, Andrea will not be watching or listening to these talks, but surveying the chat windows to find interesting questions that are critical to answer, even if they are answered later. So those will appear as YouTube comments under our video. And we did one more thing. Jessica Penn, heroically, I might say, transcribed the video from the last week and created this set of subtitles. So these videos will be more accessible to non-native speakers of English in the future, hopefully. So they can, even if due to my way of pronouncing things, critical or key terms are not very clear, the subtitles will hopefully help. And also, we decided to do something a little further thanks to the initiative by Eva, Andrea, and Emily. And we decided to put together an undergraduate Q&A. So we understand that undergraduates, high school students, undergraduates that are with us today, and they were with us last week, they may be having hard time raising their voices among all these relatively later stage career scientists. So these three undergraduate students graduate students decided to put together a Q&A based on an advice from one of the undergraduates who were in the call last week. And if you would like to attend to this, it will take place an hour before the next week's seminar for the first time. And you will need to register through this form, please. If you are not an undergraduate, or basically, if you're a graduate student or a postdoc or a professor, please don't register. But because we want this to be a smaller cohort of people who can freely ask questions, turn on their videos, discuss things, and get answers from some graduate students, and hopefully learn about these concepts a little more, even if they are not willing to ask questions during discussion sessions here. So if you are interested in this, please fill this form, and you will hear from Emily, Andrea, or Eva, and they will send you the Zoom link for next week. Okay, so this is all the housekeeping business. And now I'm moving on to a reminder of what we have done last week. Last week, we talked about microbial life. There was a brief introduction to microbial life to basically answer the question, why do we study microbes, why microbes are interesting and relevant to life on Earth? And we came to a point where we discussed these omic strategies that enable us to study naturally occurring microbial populations in a comprehensive fashion. And this week, we are going to be focusing on the power of metagenomic read recruitment, a concept that took me a long time to truly appreciate. I studied amplicon sequencing, 16S ribosomal RNA gene sequencing at the beginning of my journey at the Marine Biological Laboratory. And during that time, I had an understanding of how microbial systems were being studied, and that understanding truly contrasted with my, perhaps I should say interfered with my later understanding of general omics. So I think it is very important to understand the basics and the utility of metagenomic read recruitment, which I will try to cover. Going back to the slide, metagenomic read recruitment actually makes up a significant fraction of what's going on here. We always rely on read recruitment one way or another, whether we are recruiting reads from metagenomes or metatranscriptomes, etc. So we will get to, I hope you will have an understanding of this concept if you are not very familiar with it already. Okay, so before I go into the topic, I would also like to thank those institutions who supported this meeting, Simons Foundation and Alfred Sloan Foundation, as well as CDAC at the University of Chicago. And my name is Mehran. I'm an assistant professor at the University of Chicago. And this is our program today. So again, we have a very full program. I even added an entire section 15 minutes ago, so I'm not sure how am I going to manage to cover it without making mistakes, but we will see how that goes. Hopefully I will fit this in a single hour. If not, I'm hoping to finish it as soon as possible after the 1 hour mark. So what I want to do now is to start with the nature of the data. So I want to contrast amplicon sequencing to shotgun metagenomics. So we have a better foundation to discuss the differences and the utility of the strategy of shotgun sequencing. With amplicon sequencing, probably a lot of you are familiar with this concept if you have been dealing with microbiome data or if you have been reading microbiome stories. The way amplicon sequencing goes in a very crude fashion is like this. You have an environment with a bunch of bacterial genomes, let's say, and you use universal primers that identify a short fragment in a given genome by landing on universally conserved sites on 16S ribosomal RNA gene. This is one of the very well-established strategies in microbial ecology that allows us to amplify the information content that's in between these primer sites, which can help us identify, for instance, different kinds of organisms in a given environment. These are like collecting fingerprints from bacterial genomes in the environment. And then when we sequence those fingerprints that we amplify from the environment, we end up, for instance, learning about the existence of this genome and also to a certain degree, its abundance in the environment. And then the same goes for this genome and that genome, and you end up generating this collection of amplicons, what we call amplicons coming from this amplicon sequencing. These are, again, to remind you, small fragments coming from genomes that enable us to identify what kind of microbes they are. But primers are not necessarily, although we call them universal primers because they target a region of microbial genomes that are most commonly found to be very highly preserved, our primers are not necessarily universal. So when you use amplicon sequencing, you also end up missing organisms that may not necessarily have the kind of primer site you expect to find in bacterial genomes. So with amplicon sequencing, we can miss some microbial genomes in the environment, but apart from microbial genomes that belong to bacterial or archaeal clades of life, we also have eukaryotic genomes in the environment. So we will not learn much about their existence, or we don't, our sequencing libraries do not include reads coming from those genomes, so we don't necessarily get to measure their existence or abundance in the environment. We have plasmids in natural habitats. Plasmids are these tiny genomes per se that microbes can exchange and maintain if these plasmids confer some sort of fitness to environment stress. And we also have viruses that have a huge influence on life in certain cases. So with amplicon sequencing, we don't get to learn about these other aspects of life that may be very critical for the functioning of a given ecosystem. If we want to understand microbial life, these missed bits of information can be very, very critical. This is not to bash amplicon sequencing. 16S ribosome gene amplicon sequencing have played a critical role for us to understand the diversity of life in different habitats, and still constitutes a very significant fraction of microbiome research that is going on out there. So my purpose is not to say 16S is old-fashioned or not necessarily effective. There are certain questions that we cannot really answer with anything but 16S ribosome gene sequencing. So that is just I want to make sure that that's clear to all of you. But now let me talk about shotgun sequencing a little bit, and then highlight the major differences between them. Why would one would like to stick with amplicon sequencing even when we have access to shotgun sequencing? So imagine the same environment with the same kind of composition of different things. When we apply shotgun sequencing to this environment, what we do is basically we extract DNA from these environments as best as we can. Of course, there are limitations there as well, because when we sample a given habitat, we lose the natural gradients. When we try to extract DNA, we are not necessarily accessing to the DNA of every single organism as effectively. So sometimes, for instance, depending on your DNA extraction strategy, you may be missing information from an entire clade of life without knowing. So there are biases there, but assuming that the DNA extraction works perfectly for the smoke example, your shotgun sequencing libraries will include reads coming from all over the place of these genomes that you have been able to identify with amplicon sequencing. But you will also learn about genome sequence fragments from those organisms that you have not been able to identify with 16S ribosome gene amplicons, even though they are bacterial or archaeal, for instance. In addition to these things, you will get a lot of reads, sometimes to an annoying degree, from eukaryotic life. So for instance, if you are working with a mucosal brush, or if you swab your tongue to learn about microbial life in these habitats, with amplicon sequencing, you are going to be golden, learning all about bacterial and archaeal life in these habitats. But when you sequence the same sample with shotgun sequencing, perhaps 80–90% of your sequencing will be coming from eukaryotic DNA, because these host habitats include eukaryotic cells commonly. And not only eukaryotic genomes you will learn about through your shotgun sequencing, you will also get a lot of sequences from viruses in a given environment. And then you will also learn about plasmids that may be critical for the functioning of this system you're studying. And we call these things metagenomic short reads in general. They can be metatranscriptomic short reads as well. And it is important to note that the reason these short reads are short reads is because due to the limitations of our sequencing chemistry. that are commonly used for high throughput sequencing, such as Illumina technology. There are long-read sequencing technologies that are emerging, but neither their throughput nor their base pair quality that they can deliver currently is not really comparable to Illumina. So Illumina sequencing, short-read sequencing, is very popular. And thanks to that, we can get tremendous amount of reads from these environments that would enable us to do things that we will discuss in the later weeks. But the short reads cause a lot of problems because this is similar to this concept of generating short reads. You are basically going to a library and you want to get out all the books. But the only way you can get out these books is to shred them into smaller sentences. So you randomly shred each book and then you carry them out of the library because you cannot get the entire book out. What happens is basically you end up with a bunch of short reads, short sentences from these books that would require you to really be careful about how to make sense of this bag of sentences that are coming from all the books in the library, different editions of each book, etc. So metagenomic short read recruitment kind of helps us to make sense of that kind of data. But I feel while I was putting this together, I had the impression that one needs to highlight the fact that small things matter in these natural habitats. So amplicon sequencing is extremely affordable and has the luxury of very well-established protocols that anyone almost can use this strategy to study microbial systems. And there are many computational tools that help us to make sense of such data. So there are many advantages, and which enable us to characterize large number of habitats all at once. On the other hand, shotgun sequencing has the advantage of characterizing those genetic components of a given environment that are not necessarily recognized by our primers, which are forever going to be limited with their access. But why should you even care? Why should anyone care about viruses, for instance? Viruses that don't infect us, that infect bacteria. So, or plasmids, these tiny genomes that carry 1 to 100 genes or more at a given time. So why should we care about this? I have this small story for you to highlight. So we're going to take a little side track now to talk about why small things matter. And I'm going to share with you one of the stories that had a huge influence on my thinking about microbial life and in general evolution on Earth. And this story is coming from a relatively well-established field that studies host microbe interactions in the context of this mosquito, Culex pipiens, and this microbe that is called Wolbachia. So this guy is one of the most effective vectors of some of the worst human diseases, so including Zika and dengue. So the population size of these mosquitoes in a given habitat is directly correlated with the rate of infection among humans, because they are very effective vectors of these diseases. Why do we care about their population size? Because we learned that this bacterium, in Seth Bordenstein's word, there's a pandemic going on Earth, that Wolbachia infects more than 40% of arthropods on Earth. Arthropods include a lot of things that we very well know about, from butterflies to wasps to bees to mosquitoes. So it's the most populated from a point of view of species names in the tree of life, part of eukaryotic life. So 40% of arthropods mean a lot of different kinds of eukaryotic organisms. And Wolbachia successfully infects 40% of arthropods. This is an intracellular bacterium that lives in the ovaries of Culex pipiens. So essentially, this bacterium lives in the ovaries of this Culex pipiens and is being vertically transmitted from one generation to the other. So what is interesting about this is this. When a female mosquito and a male mosquito comes together and have sex, they create all these offsprings. Well, this is how things go in nature. We know that. if there is a female mosquito with Wolbachia, because Wolbachia is not essential for the survival of the mosquito itself. But you will see how it becomes relevant for the population size of the mosquito, and how the ecology of Wolbachia is partially defined by the ecology of these mosquitoes. When a female mosquito is infected by Wolbachia mates with a male mosquito that's infected by Wolbachia, I guess it wouldn't be surprising to you to see that the offspring, all offspring, has Wolbachia. Every member of the resulting offspring will have Wolbachia because it is vertically transmitted from mother to infants. And in the case of this situation where only female mosquito has Wolbachia, it is still, it results in a Wolbachia infested litter. So all these guys still carry Wolbachia. So you may be asking right now, so what happens when the female does not have Wolbachia, but if Wolbachia-less female mates with a male that is coming from this litter, for instance? This is the fascinating part. You get no offsprings whatsoever. So these mating of this kind causes this phenomenon called cytoplasmic incompatibility, and there is no offspring. Something interferes with the DNA replication in the egg, and things go wrong. And even when you have, this is more recent research, different strains of Wolbachia, let's say for now, you don't get offsprings. So there is this interesting phenomenon going on in nature, and you can see where the interest lies when you realize that if different strains of Wolbachia can lead to no offspring, despite the mating behavior, you can control the population size of mosquitoes out there by tinkering with which Wolbachia they are infected with. So you can imagine biotechnological applications of such understanding, right? So the model is essentially what I'm describing you here so far is that there is this mosquito and there is this bacterium that infects this guy and influences its reproductive behavior. But actually things got much more interesting when you realize that there is a phage that infects Wolbachia. So this story does not end here. I think so far it's already very interesting and fascinating, but it continues to be even more ridiculously interesting, because you realize that the system also includes this phage that infects this Wolbachia bacteria. And if you go to the literature and read from Seth Wardenstein's group, they have been able to characterize the mechanistic underpinnings of this cytoplasmic incompatibility. And they have been able to show by doing these genius experiments, putting the genes from the phage to the genome of the mosquito and removing all the phage and bacterium from the equation to show that there are two genes encoded exclusively by the phage that infects the bacterium that causes cytoplasmic incompatibility. So what I'm telling you is that in this model I just showed you, the influence of Wolbachia is basically simply carrying around the phage. So it is not the bacterium itself, actually. It is the phage that defines the evolution and ecology of this eukaryotic host. So this is so interesting because it tells you that there is this organism living out there that we had known about much, much longer than we knew about Wolbachia and its phage. And we had been curious about its ecology and evolution. And yet, this organism is infected by a microbe that is infected by a phage. And the phage, this puppet master who evolved a very long arm, in fact defines the reproductive behavior of Culex pipiens. So this tells you that if you are a naturalist studying the ecology, evolution of these mosquitoes, and if you completely miss the existence of Wolbachia, or most important in this case, the phage, then you are going to come up with all sorts of stories about what influences the population dynamics of Culex pipiens. And the funny thing is, nature is filled with chaos, especially at this level of biology. So you will find stories. If you go back to maybe 100 years back and read about what scientists at the time thought about the ecology and evolution of Culex pipiens and sexual behavior, perhaps you will find very compelling stories completely excluding the existence of this this influence on its behavior. So apart from the fact that this really makes you think to what extent evolution on Earth is influenced by these kinds of unexpected and surprising phenomena that we are just learning about, this to me also shows the importance of being able to characterize an environment as a whole, rather than just, let me say, it highlights the importance of also keeping in mind that we may want to ask some questions to the total environment rather than amplicons. So going back to this, I hope this somewhat elaborates a little more. Why should we, how can we find ourselves in situations where we realize that we may need shotgun sequencing of the environment rather than amplicon sequencing if we want to understand microbial systems better? So, okay. So we end up with these metagenomic short reads when we do shotgun sequencing. As I said before, this also could be metatranscriptomic short reads, and everything I'm going to tell you downstream is going to be applicable to metatranscriptomic reads as well for this week. And the one thing that we need to understand is the concept of read recruitment. So what do we even do with these things? Some, okay, just to give you a brief, just a brief note, there are many ways to make sense of these metagenomic short reads without any read recruitment, the way I'm going to describe it anyway. For instance, you can take these short reads, you can blast them against existing genome collections, and you can annotate individual short reads with taxonomy, or you can annotate individual short reads with functions based on which genes they match in known databases. But and then you can count these numbers up, and then you can come up with certain, you can come up with an exact spreadsheet. This environment has this function, this percentage versus this environment. Next week, we are going to talk about limitations of those approaches and how they can lead to misleading insights. But this week, we are going to discuss how do we do read recruitment? What do we do with these kinds of data? So the next chapter of our conversation will hopefully give some insights about that. Going back to these metagenomic short reads you get from an environment. You did metagenomic sequencing, shotgun sequencing, you have all these short reads. One thing you could immediately do is that if you have a genome, let's say you have a genome, and you expect this genome to occur in this environment. Let's say you are studying Streptococcus, a Streptococcus organism in human oral cavity, or a Bacteris fragilis organism in human fecal matter, or SAR11 in surface ocean. So you have a genome or a plasmid genome. It doesn't really matter at this point. What you can do is to apply read recruitment to interrogate whether this genome represents a population in the environment or not. And the way read recruitment goes in the most crude fashion, if I try to explain it, is this. So we use various software tools to accomplish this task indeed. And what they do essentially boils down to the notion that the tool for you takes a look at the genome and identifies a short read that matches to this genome, to this part of this genome, let's say, and aligns that short read to here. And then that short read, in most frequently used way of studying these metagenomic short reads, we usually don't align the same short read to multiple contexts. So we take it out of, let's imagine we take it out of our collection of short reads. And then next, we identify another short read. We look at the genome, and it turns out that it matches to this part of the genome. And then we align it there, and we find another one, and so on and so forth. So we start aligning things to our genome. And at the end of the day, when this is done, basically, you collect all the reads that match this genome from this environment. So basically, this gives you the power to detect, for instance, be able to speak of the existence of this genome in this environment. So we will go into details of this a little more, but essentially, even when you don't know these colors, obviously, when you have no idea about the content of this environment, you still can come up with your genome and ask the question, I want to understand if this particular environment with these reads contain my genome. So that's the beauty of this. You do not necessarily need to characterize short reads beforehand to be able to ask the question if my genome is there or not. For instance, let's imagine you are working in a lab. Let's say you're working in an immunology lab, and you cultivated an organism from the human gut that seems to have some properties that makes the immune system realize its existence in this environment. And let's say you found out that presence or absence of this genome in a mice model, let's say, in a mouse model, influences how the immune system behaves. Let's say, so what I'm trying to tell you is that let's assume you have a genome that seems to be key for understanding disease. So one of the things you can do right then is to gather a bunch of human gut metagenomes or mouse metagenomes from different facilities and ask the question, if this genome that I associate with disease occurrence very significantly, whether it occurs or not in other healthy gut metagenomes of mice coming from labs where people had not been studying this disease. For instance, this strategy, by just downloading those metagenomes and using your genome that you cultivated in your own lab, would enable you to immediately answer the question to what extent this population is present in other metagenomes. So this is the beauty of it. And in fact, this is not something you could do with amplicon sequencing, because if you were to use a very small fragment of the genome to answer this question, because the diversity of the rest of the genome for almost all populations will be vastly more complex than the diversity of that small thing you're looking at. It will be like, okay, I met this person who had a driver's license from Louisiana and told me very good stories. So now I'm going to go to a city and collect all the people with Louisiana driver's license, and I will expect them to be able to tell good stories too. So relying on that small bit of information, while it does not allow you to make very highly resolved conclusions about the things you are trying to understand, metagenomic read recruitment using larger context of genomes is very effective to track populations. So I hope the term population will start meaning something to all of us in a similar comparable fashion towards the end of the seminar series. But I use this term very with purpose. Okay.
Marin, could I stop you for a second?
Yes.
There's a question from the chat, which I think is important. Simone is asking, would the read recruitment approach be good to quantify prevalence of a genome or population in a sample?
Prevalence, not abundance. Is that correct?
Prevalence.
Okay, yes, I think that's a very fair way to use the metagenomic read recruitment. And let's talk about that a little bit. So prevalence is, the way I understand prevalence is the existence of a genome in a given environment, being able to confidently say this genome represents a population that is in this environment based on these read recruitment. And another term we commonly use to describe microbial populations and their composition is abundance. Or you can say this genome is more abundant in this environment compared to this environment. So there are ways to, while all of them require a lot of care and attention, there are ways to address both of these through read recruitment. So and these are the obvious characteristics, the way I define, of microbial populations in the environment that we can learn through read recruitment. So now I want to focus on these statistics of the data. So I again will use an example and say, let's say we have a reference context like this. The reason I'm not calling this anything more specific than reference context is because this shall not tell us anything more than the fact that this is just a piece of sequence. This could be an entire circular genome, or this could be a small plasmid with 2 genes in it, or this could be a single gene that you're interested in, or this could be an entire eukaryotic genome assembled into 200 contigs. It doesn't matter. It's just a sequence. But for the sake of this argument, let's say this is this cartoon represents a bacterial genome with multiple genes in it. Okay, and assume that we have an environment from which we generated metagenomic reads. So essentially, a collection of reads that match that colorful mishmash of everything in the environment. And we can do read mapping, we can do read recruitment to learn about the organization of the short reads in comparison to this genome. If you remember, I mentioned one of the utility of the read recruitment is to be able to say, this short read here in my metagenomic sequencing results matches to this part of this genome. Okay, so when you, and we have specific file formats, we can talk about them later. I'm hoping to put together a seminar series on practical matters and actually doing these things with real genomes and real metagenomes. But until then, I hope we can simply take it as a fact that there are tools that enable us to do these things. There are file formats that enable us to characterize this data like this. It would take you basically a relatively small amount of time to get from a sequence you have and metagenome you have to this point. So to look at how these short reads match. And basically, just to reiterate, metagenomic read recruitment will find all the reads that match to different parts of your genome. And we can display them like this. This is one of the common strategies you perhaps saw before or used. For instance, what do you think the blank area mean? It makes probably more sense if I put these additional information here. Let's say this is our genome and these are the genes. And then you recognize that the missing reads here, for instance, correspond to this set of genes, 2 genes here. So, for instance, one thing one can immediately recognize from this is that you have a reference sequence, you have a reference genome, and you have an environment that contains a population that is similar to the reference genome you have. But of course, the genome you have may not be identical to the members of this environmental population. And this tells me, when I look at this, for instance, there are 2 additional genes in this reference genome that is not necessarily a part of the population, the members of the environmental population commonly carry around. So this could be a hypervariable genomic island, or this could be a phage-mediated insertion of some genes that are not critical for the survival of the environmental population, and so on and so forth. There may be many different ways to investigate this, but once you see this, then you can say, I want to take a look at the functions of these genes to be able to say more about their relevance or interest to my own study. So that's why it is important to be able to see these patterns through metagenomic read recruitment at this level of resolution, rather than summarizing the entire read recruitment into a single mean coverage quantity, which is what is commonly done in the field for practical purposes. But what I want to try to communicate to you is that metagenomic read recruitment holds a lot of power if it's used carefully. And there is another story here. Perhaps you are recognizing that there are these bunch of genes that seem to be covered much more than the rest of the genome. So let's discuss about what would we expect if everything worked in the simplest fashion we can envision them. Imagine you have an environment that contains 100 copies of this one genome you are holding in your hand. If you were to, this means, okay, let me use our library analogy. Let's say there's a library and contains 100 copies of this book you are holding in your hand outside. And there's nothing else in this library, to simplify things. As we spoke about moments ago, there is no way we can get these books out of this library as a single piece. So what we do, we give these 100 books to people and ask them to shred them into smaller pieces. So they randomly shred these books into tinier sentences. It doesn't have to be complete sentences, but let's say 5-word long fragments of the book. And then they bring all those 100 books shredded in a bag. And then we are basically going through this bag, taking out a sentence and comparing it to the whole book we have. And now we start counting. Sorry, my dog is very excited about this library analogy now. We're counting the number of times we observe a given sentence based on the number of fragments that match the sentence. So in an ideal world, you would expect every section of this book to be covered by about 100 fragments. Because if you align the shorter fragments in the book like this, because there were 100 books, you would find out that the entirety of the book is basically covered by this many fragments. So in what situation this could happen? For instance, if you have an edition of the book that has an extra paragraph that did not appear in the previous editions of the book, then you wouldn't see any fragment coming out of the shredded library bag to contain that part. And similarly, we can discuss using the same ecological thinking to discuss here, for instance. What might be going on here is there are multiple explanations for this part as well. For instance, it could be the case that this part of the book is now occurring twice in the edition in the library. So there could be a duplication event for the environment population, where you find the occurrence of this particular genomic region to be non-unique anymore to the genome. Or in a more complex setting, you could have another set of books that are vastly different from this book, but they share this paragraph. So maybe we're talking about some conserved functions that are very essential for the survival of these populations, the environmental populations in the environment. So these 3 genes may be occurring in multiple populations. So read recruitment will tell us that based on this phenomenon we see here, that these are not like the rest of these genes. Or, for instance, this could be a phage, a prophage in the genome, a virus in the genome that is being expressed. So there are phage sequence in the genome, and there are phage particles carrying this sequence in them. When you extract DNA and recruit reads, maybe you will see that kind of thing. So again, what I'm trying to tell you, we had been talking about this single cartoon all these minutes. Imagine the amount of things one could discuss by looking at real data and coverage values of their genomes across metagenomes. Okay, so let's assume we have more metagenomes than one, and let's say we have 3 environments. The beauty is that we can put the same reference sequence we have in the context of multiple environments. And I'm just going to highlight these sections because those were now genes of interest that we had been talking about them so much. So let's keep them in mind. And let's say in metagenome 2, this is the kind of coverage we get. So this is how short trees, fragments of environmental genomes match to this context. So in this environment, you realize that the entirety of the genome is detected, is present. So talking about presence, now you can confidently say, regardless of how much of this guy is in this environment, my reference sequence does occur very likely in this environment as a whole. And this gives you an appreciation of the presence of this genome at the level of resolution of individual genes. For instance, now these 2 genes all of a sudden seem to be in this environment. So by looking at these gene level characteristics, you can start making sense of differences across environments as a function of the collection of genes you have access to through your genomes. And for the sake of the argument, let's say this is your environment 3, and this is how reads match to your genome. What would this tell you? This would tell you that the reference sequence you are looking at in this example, a bacterial genome, let's say, is not necessarily in this environment. So, but these 2 genes that carries must be encoded by another genome. This is almost like you have a cookbook, you're standing in front of a library, and you ask people to shred the books inside, and you get out everything. And then when you match those shredded pieces, you realize that only one recipe occurs in some of the books inside, because it's a well reserved, commonly relied upon recipe for a bread, for instance. So now you know your cookbook is not in that library, but these 2 genes represent, there is a population that encodes them. So so far what we had been talking about can be boiled down into two metrics, two statistics, very simple statistics. One of them is coverage. I already mentioned multiple times. For instance, the coverage is the depth of basically the coverage in a more formal way. The average number of short reads that map to a single nucleotide position across the entire reference context. So basically you are going nucleotide by nucleotide and counting the number of reads you observe when you go down like this, and you average that number out. For instance, let's say the coverage of this genome in this environment is 10x. We can talk about the utility of this actual number and what does it mean, but for now, let's assume that this simple statistic simply tells us the depth of the number of short reads that are matching to this context. So imagine, going back to the library example, this would tell me that there are about 10 copies of this book in the library. So that's the utility of this. And with the same line of thinking, the coverage of this genome in this metagenome would be, for instance, 3x. So now comes the interesting part. What do you think the coverage of this reference context in this third environment? You probably will be like, yeah, perhaps 2 or 3 when you look at the average coverage. And the point I'm trying to highlight here is you may find the average coverage of a given genome to constitute a reasonable, perhaps remarkable number, even though the population is not there, and yet recruits reads given only a single fraction of it. So for instance, to example this a little further, you may have a genome that carries a phage, a prophage, and this prophage may have infected another population that's extremely abundant in this environment, but has nothing to do with your genome. And if this population is abundant, or if the phage in the environment is in lytic life cycle, you will get a lot of matches from that environment to your genome. And if you're not careful about how the read recruitment looks like, then you are going to mistake that your genome is equally abundant in environment 2 and environment 3, when in reality we are seeing a very different, two very different ecological phenomenon taking place. So the way we recover from this is to use another simple statistic. And I'm sure you can envision what else would you add to this to make it a little more clear to what extent a given genome can be described with simple statistics in a given environment. And we call it detection. So the detection metric is essentially telling us the fraction of the genome that is covered by at least one short read. Of course, this is a very loose way of actually defining something such so important, because you may, for instance, have what are we going to do if the genome is covered 10,000x everywhere, but here only 1x? Are we still going to say it is detected? And I would argue, yes, we are going to say it's detected, because detection does not care about abundance. But from another point of view, you can also assume that if the genome is not 10,000x covered, if it's 1,000x covered, you may not have enough sequencing reads to detect this portion. So again, all these things I'm telling you are very important considerations to think about when you're studying your environments through read recruitment. We have to be aware of alternative explanations of the ecological phenomena we observe through these statistics. And in this case, for instance, detection would be 1.0, because the entirety of the genome is covered by at least one short read throughout. And in contrast, in this case, the detection would be 0.2. So if you are paying attention to both of these statistics, then you would be able to say, okay, the coverage is identical, but detection tells me that only a small fraction of my genome is detected in this environment number 3. So let me take a better look at it, for instance. I hope this part so far makes sense, that these are the things that are so obvious that we can see immediately through read recruitment when you have a reference sequence in a bunch of metagenomes at a resolution and at a level of confidence that we cannot achieve with amplicon sequencing. So tracking microbial populations across environments through amplicon sequencing is a very crude way of understanding their actual occurrence and the manifestations of these populations at different environments that may render us unable to answer some critical questions for the ecology of these populations. But shotgun metagenomic sequencing in read recruitment helps us to observe such things at a very high resolution. I hope that part is clear. This is the obvious things we can discuss when we use metagenomic read recruitment.
Maren, sorry.
Yes.
Just it seems like there's confusion from the audience.
Yes.
Could you just go over one more time the difference between coverage and depth?
Coverage and depth essentially mean the same thing. Coverage and depth, or coverage and detection?
Coverage and depth.
Okay. I am very sorry for the confusion. I try not to use multiple terms to describe the same thing, but sometimes it happens. The coverage, also called depth of coverage, is the depth in this sense, depth of sequencing, the depth of these sequences when you stack them, when you align them to your reference context through read recruitment. Basically, how many, as you go deeper and deeper, if it's okay to give a geometric reference, if you go deeper and deeper after matching your short reads to this context, when you ask the question, how many of these reads do I observe as I go deeper and deeper, and take the average of that number across every nucleotide position throughout the genome, you have the coverage. You have the depth of coverage of the genome, or the coverage of the genome. So they're essentially, they mean the same thing pretty much. I hope that is clear now, a little better. And detection. So if you need to, again, I always think in shapes. So if it helps, you can think of coverage as a vertical property, and detection as a horizontal property of these kind of data. All right, so now let's move on to seeing the subtle through metagenomic read recruitment. So as I mentioned, we talked about how we can detect the prevalence or even abundance in some cases, although I would really like to take a couple of minutes in sometime next week to discuss how it is nearly impossible to talk about quantities through omics data. We cannot, I believe we cannot quantify abundances in a very convincing fashion by using omics data. I will skip that for now, and I will say basically the obvious things we can learn is abundance and in presence of any given sequence, whether it's a bacterial genome, viral genome, or eukaryotic genome in a given environment. These are the obvious things. Subtle things is going to be equally important for some other research questions. For instance, let's focus on this part of this single gene, and imagine that as if we are able to see these individual sequences here. When we look at the read recruitment at the resolution of these nucleotides, we can see things that we may not be able to see by looking at overall patterns we observe through read recruitment here. For instance, let's say for this one nucleotide position, you look at every short read and the nucleotide in each short read that matches to this location. So this short read, well, the lengths do not scale, I'm sorry. For instance, this is supposed to be 100 to 250 nucleotide long short read, but here it's mapping to only 2 nucleotides. So I want you to ignore that and assume what we're looking at still does make sense. When you go to every short read that's aligned here, for instance, this short read is aligned here by the mapping software or read recruitment because it is an A and T. So this is AT. And this one is aligned here because it's an AT2, etc. This one is aligned here because it starts with the T, and we don't know, maybe it's a C here. So, and so on and so forth. And if you go through this column and see that here there's a read, instead of a T, there's an A, what would you think? You would think that sequencing is not an error-free process. Therefore, maybe there's one short read in my sequencing results that contains an A when it was meant to contain a T. So that was a sequencing error. But what if you see that A is occurring a little too frequently for you to feel like this could be due to sequencing error. Let's say this is happening 50-50. 50% of the time, short keys that match here contains a T, and another 50% of the time, they carry an A. So why, how would you describe that? How would you explain this phenomenon? Why is this happening? This is basically saying that you have a cookbook, you are in front of a library, and everything is shredded and carried out, and you are matching fragments, and you realize that 50% of the time, this one sentence uses a different word. So the sentence, everything is the same, but this just single word changes across mapping results. 50% of the time, you see this word, and 50% of the time, you see the other word. And it would probably make you think that perhaps there are two different editions of this book in this library that occurs in about the same number of copies. So let's say in the second edition, the author realized that there's a better word to use, and they chose that. And therefore, now you're shredding them all, you're seeing this. This is essentially trying to focus on microbial population genetics to make sense of environments at the level of resolution that could enable us to discuss different editions of books and so on and so forth that we can't see from here, and answer the question, well, this genome is here, that's brilliant, but does every member of this environment really looks like this genome at every gene? You may be able to identify certain things that are critical for the survival of some subsegments. For instance, you may realize that that word between these two editions of the book influence which communities decide to not read the first book and choose the second edition of the book, for instance. Or you may find out that there's a very significant biogeographical distribution of these different editions of the book. Some states only read this version, and some other states do not understand that word because their cuisine is different. So this, of course, would improve your understanding of the state you're studying. So it is, in some cases, very critical to follow these trends. And if we have time, we are going to talk about the tools to study these microbial population genetics phenomena. But this is a very rapidly emerging field that many people who study omics pay attention to. And for instance, you can say that there are two editions of the book by only looking at this. But what if you look at a different nucleotide position and you see this, 80% to 20%? So now you're realizing that, okay, that perhaps this is not only two editions of the book, but perhaps there are three editions at different proportions, or it has to be at least four in this case. So this kind of information can be used to even try to estimate the number of, let's say, haplotypes or different subpopulations in a given environment. And we commonly use those things to discuss to what extent can we attribute this kind of heterogeneity to improved fitness as a function of environmental change. This is one of the most important questions that people try to answer in our field. To what extent these variants can lead to better fitness under what circumstances, and which genes are most critical, which genes are, for instance, never change. If you see multiple editions of the book, every sentence is changed a little bit. But if there's one sentence that always the same across all the editions, it may tell you that while this particular sentence cannot change, it's so critical. In evolutionary terms, the prefiring selection ensures no heterogeneity there. In the book analogy, this sentence is so critical for this book to communicate to its audience, it cannot be changed. There are things we can learn from environments at this level of resolution. Finally, let's say you have a nucleotide position like this. You have a genome, but everything is an A here instead of a G. So you observe 0% of G and 100% A. And this can happen as well. This would mean that the environment is relatively clonal. So all the editions in the library, all the books in the library have changed this word, and there is not a single copy of the edition you're holding in your hand. It wouldn't mean that the edition you have in your hand is useless or cannot be read or cannot be found anywhere, but you would know that this particular library contains 0 of my edition, but it's pretty consistent that everyone agrees that this word is the word to go with. So there are all these things we can learn and dissect by only looking at read coverage data. And it's an extremely powerful way to characterize things at the nucleotide or gene or amino acid or codon level, to study ecology of our organisms through obvious statistics and their evolutionary pressures they have to deal with, or environmental parameters that force certain genes to behave different than others through these kinds of statistics. I hope that's somewhat clear. I hope so far this gives you some insights into the power of metagenomic recruitment, as the session promised in its title. Finally, I want to talk a little bit about genome-resolved perspectives. So far, I've been telling you that the reference context can be anything, but I think it is most commonly is a genome. So the most frequently we most frequently use genomes to do metagenomic recruitment, and the way I think about this is twofold. Genome-resolved metagenomics includes those shotgun sequencing of a given environment, getting all these reads, and then using these metagenomic shortlists with reference genomes that already exist. So for instance, imagine what you could do with all the genomes people have isolated from the human gut, and with all the metagenomes out there that are generated from the human gut. You could use metagenomic read recruitment from all these existing metagenomes to those genomes that you have access to characterize each one of them. You can talk about what fraction of this genome is distributed across metagenomes, and which genes are in everyone when this population is present, and which genes are only in a subset of people. Are those subset of people coming from the same country, coming from the same disease backgrounds, coming from the same community type? So there are so many different questions one can ask with reference genome. So by recruiting, for instance, here I type mapping, and this is again read recruitment, but you will also hear this in the field as read mapping. When you recruit reads from this metagenome using this reference genome and that reference genome and exhaust all the reference genomes you have, you are going to learn a lot about them, indeed. But then, as in this simple cartoon tries to demonstrate here, that you are going to have a lot of reads left in your metagenome that is not necessarily characterized by anything. Because as we know, we are unable to, using our commonly used strategies to grow microbes, we're unable to grow every microbe from every environment. There are many microbial populations that are yet to be characterized in our culture collections. And you can imagine that even when we characterize them in our culture collections, there will be many different manifestations of those members we were able to bring into cultivation. It is like saying an alien comes to Earth, captures one human being, and then they live the next 1,000 years and in their biology books describing human behavior and thinking and emotions using that single human. It's not going to be representative. And for a microbial world, it is not much different than that. Even we have cultural representatives for every single organism out there, this is not going to be enough to characterize environmental populations. We will still want to understand gene level patterns, evolutionary processes that are acting upon different parts of this genome across different environments as a function of environmental change. So well, I'm not saying this is not a viable strategy. This is an amazing strategy, and there are many papers from the last few years that people have characterized metagenomes using reference genomes, and I hope now, if you ever go back to those papers, these things will will help you a little more to make sense of what they have done, and you will keep seeing the coverage, the detection values, even though detection is less discussed in the field as of today. And you will get to appreciate that, well, did they look at individual genes, their distribution, etc. Okay, so, and probably most of us are aware in this crowd that another way of making sense of these metagenomic short reads, in addition to the alternative of making sense of them through reference genomes, is to assemble them. I am not going to go into details of this today because this is an entirely different discussion topic that will take place next week, actually. De novo assembly, essentially stitching together short trees that are partially overlapping in a very crude way, if I have to define. And generating longer contexts is a way to describe genomic contexts that are longer than our short reads. So this is, I know how silly this sounds when you think about it. Basically, you're getting all these books in this library shredded in front of you, and then you're trying to reconstruct the book itself because you simply cannot get the book out as a whole. And this is rapidly changing, luckily. Long read sequencing enables us to work with people who shred books into much larger fragments. They're able to keep in mind many more words than our short read sequences, and then it becomes much more easy to reconstruct the book, as you can imagine. But the problem there is that the throughput is low. Therefore, to what extent we characterize books that are occurring in small quantities in the library becomes a question, etc., etc. But going back to this, we can also assemble these metagenomic short trees into contigs. The word contig essentially means contiguous DNA segment, and basically gives us a better understanding of the genomic context that helps us to do multiple things, including metagenomic binning. So what you do, essentially, we do not necessarily assemble a single book into a single book. When we use even all the short fragments coming from a single book, we end up with more than one pieces of that book. So when you're thinking about a library, you are going to end up with many more contexts than the number of actual microbial organisms there due to some bioinformatics artifacts we will discuss next week. But there are strategies that have been pioneered by the Banfield lab years ago, and now we commonly use through new computational strategies and have been applying it to every environment so far. It enables us to identify those pieces of genomes that are fragmented and put them together as metagenome assembled genomes, or MAGs. You may have heard of that in recent papers, and study them just like the way we use reference genomes to map these short trees back to, again, talk about those properties of these organisms. Now we gained access to their genomes without having to go through cultivation. So as you can imagine, and if you've been following the field, this way we can increase our coverage of genomic understanding of environmental populations without having to grow every single bug in the lab environment, which is extremely powerful, but comes with significant caveats, of course, such as the fragmentation of these genomes, not knowing exactly how the synteny looks like, and the fact that we are going to be unable to assemble parts of these genomes that are shared across multiple organisms in a given environment, and so on and so forth. We are going to discuss these things in a little detail next week. But even when we're able to assemble our metagenomic short trees, which is not necessarily applicable to every single environment, same level of efficacy, unfortunately, and even when we're able to bin our genomes, contigs into individual metagenome assembled genomes and recruit these reads, there are going to be some reads that we're unable to characterize in our metagenome. So those may be coming from organisms that are low abundance, so we couldn't assemble, we couldn't bin. They could be coming from certain genomic regions, like I intentionally put these blue reads here, because even though we have a MAG that is represented by color blue, there may be regions in this genome that is not well assembled, so therefore left behind. And there are going to be reads that are not fully characterized. And the question, once you assemble your metagenome, once you characterize your metagenome through metagenome assembled genomes, what fraction of the metagenome itself is mapping back to this is a very important one. That is, again, mostly missing in most studies. So knowing the fraction of the reads here that is not characterized by these genomes is an important one. But also, we have to keep in mind, for instance, you will never be able to assemble the human genome through metagenomic short read sequencing. So if you have unknown eukaryotic organisms in your sample, you are going to see higher percentages of reads uncharacterized due to the fact that this workflow does not necessarily work well for eukaryotic genomes. But still, we need to, I think about these things comprehensively and kind of know why are these things happening. Okay, I hope this part somewhat clarified that the genome-resolved characterization of metagenomic sequencing involves both reference genomes and genomes that we are able to reconstruct directly from metagenomic short trees themselves. And next week, I'm hoping to focus on key concepts that enable us to reconstruct these genomes from these environments and discuss that process and its caveats and its advantages and etc. In a more detailed fashion. With that, I thank you very much. Again, if this is your second week, thanks for coming back. I'm surprised that you are here again. And if it's your first week, I hope we'll see you again. And I thank the members of my group who have been very supportive of this endeavor that seems to be taking much more time than I had envisioned and anticipated at the beginning. And as usual, those who supported our science so we can do these things.